18. Classification, biodiversity and conservation. Classification is the grouping of organisms on the basis of their similarity and differences or according to their characteristics and evolutionary relationships with one another. All types of living organisms found at an ecosystem is called biodiversity. Conservation refers to the process of saving endangered species to maintain biodiversity and natural beauty of the biosphere. For the above-mentioned purposes, field trips of local, national, and international levels may help the students to recognize, classify and conserve different organisms that are going to be extinct in near future. Different strategies like conservation of natural habitats, botanical gardens, zoos, seed banks, reforestation, artificial methods of, new techniques of reproduction, etc. should be adopted for the continuity of smooth life of all the creatures of this planet. 18.1 Classification. Classification is a system used to help understand the diversity of living organisms, by placing organisms with similar characteristics into groups for easy reference. The binomial system of nomenclature is used, with each organism given a two-part Latin-based name, e.g. Lumbricus terrestris, earthworm, or Primula vulgaris, common primrose. The first part, generic name, states the genus or group of closely related organisms. The second part, specific name, states the species. This is unique to the particular organism. Taxonomy is the process of ordering groups of organisms sharing common features, called taxa. For example, humans are in the phylum Chordata and class Mammalia. Taxonomy is the process of ordering groups of organisms called taxa into hierarchies, which attempt to account for their evolutionary origins. The taxa are groups of organisms sharing common features. There are 7 levels, with a kingdom the highest and a species lowest, as indicated below with the classification of species of seaweed, bladderwrack, and humans. 18.1 to 1 Species. A species can be defined as a group of organisms with similar morphology, behavior, physiology and biochemistry that are capable of interbreeding to produce fertile offspring. However, in practice, it is not always possible to determine whether or not organisms can interbreed. Comparisons of the DNA nucleotide sequences of 2 species can give information about how closely they are related to each other. 18.1 to 2 3 Domains of classification, archaea, bacteria, eukarya. Species are classified into a taxonomic hierarchy comprising domain, kingdom, phylum, class, order, family, genus, and species. There are 3 domains: bacteria, archaea, and eukarya. Comparison between prokaryotic and eukaryotic genome. Prokaryotic and eukaryotic genome has some similarities such as 1. They both consist of DNA that forms chromosomes in the cell. 2. Each chromosome consists of a single DNA molecule with attached proteins. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046104457. 88. 3. They both contain genes that are responsible for the coding of proteins and the organism's inherited traits. 4. They both can undergo replication, where each replication process of a DNA molecule produces 2 DNA molecules. The differences between prokaryotic genome and eukaryotic genome. Prokaryotic genome, eukaryotic genome. 1. The DNA content in each chromosome is less. The DNA content in each chromosome is more, so the length of the chromosomes is longer. 2. Each chromosome consists of circular DNA found in a nucleid. Each chromosome consists of a long and linear DNA that is not found in a nucleid, but found in a membrane-bound nucleus. 3. Each prokaryotic cell usually has only a single chromosome, thus has single DNA molecule. Each eukaryotic cell has multiple chromosomes, thus has a number of DNA molecules. 4. Circular DNA forms plasmids. No plasmids are formed. 5. DNA does not contain introns. DNA contains introns. 6. As there are no introns, there are few regulatory sequences needed. There are more regulatory sequences due to the introns. 7. DNA is transcribed into mRNA and used immediately in translation. After transcription, pre-mRNA has to be processed and intron splicing occurs before the mRNA is used in translation. 8. There is no compartmentalization of transcription and translation process during protein synthesis. There is compartmentalization in the cell which enables transcription to occur in the nucleus and translation to occur outside the nucleus. Archaea and bacteria are prokaryotes. Archaea differ from bacteria in the lipids in their membranes, their ribosomal RNA, and the composition of their cell walls. All eukaryotic organisms are classified in the eukarya, which is divided into 4 kingdoms: Protactista, fungi, plantae, and animalia. 18.1-3 Archaea versus bacteria. Archaea is a domain of single-celled organisms. These microorganisms lack cell nuclei and are therefore prokaryotes. Archaea were initially classified as bacteria, receiving the name Archaebacteria, but this term has fallen out of use. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 89. 18.1-4 Taxonomic hierarchy. Taxon description: Bladderwrack Fucus vesiculaceus. Human Homo sapiens. Kingdom: largest group of organisms with common features: Protactista, Animalia. Phylum: major subdivision of kingdom: Phaeophyta chordata. Class: group of related orders: Phaeophysi, Mammalia. Order: group of related families: Fucales, primates. Family: group of closely related genera: Fucaceae, Hominidae. Genus: group of related species: Fucus, Homo. Species: group of organisms capable of interbreeding to produce fertile offspring. Vesiculosus sapiens. There are 3 different species concepts. A biological species is a group of organisms that have many features in common and are able to interbreed and produce fertile offspring. A morphological species is a group of organisms that share many physical features that distinguish them from other species. An ecological species is a population of individuals of the same species living in the same area at the same time. 18.1-5 Kingdoms. The 5-kingdom system is the current scheme of classification and recognizes the kingdoms listed below. Kingdom characteristics examples. Prokaryote lack nuclei and other membrane-bound organelles. DNA is circular. Escherichia coli anabaena. Protactista eukaryotic organisms with envelope-bound nuclei, neither fungi, plants, animals, often unicellular or assembled from similar cells. Amoeba, green algae. Fungi, non-photosynthetic eukaryotic organisms, non-cellulose cell, lack cilia and flagella. Muca, bread mold, agaricus, mushroom. Plantae, multicellular photosynthetic eukaryotic organisms, life cycle includes a diploid embryo stage. Mosses, flowering plants. Animalia, multicellular heterotrophic organisms, nervous coordination. Annelids, chordates. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 90. 18.1-6 Viruses. Viruses are not included in this classification because they are acellular, they have no cells and are composed of only nucleic acid, DNA or RNA, surrounded by a protein coat. Viruses are classified according to the type of nucleic acid that they have and whether it is single-stranded or double-stranded. 18.2 Biodiversity. Biodiversity is all the different kinds of life you'll find in one area, the variety of animals, plants, fungi, and even microorganisms like bacteria that make up our natural world. Each of these species and organisms work together in ecosystems, like an intricate web, to maintain balance and support life. Biodiversity is considered at 3 different levels: variation in ecosystems or habitats, the number of species and their relative abundance, and genetic variation within each species. 18.2-1 Ecosystem and niche. An ecosystem is a relatively self-contained, interacting community of organisms, and the environment in which they live and with which they inherit. A niche is the role of an organism in an ecosystem. It is how it fits into the ecosystem. 18.2 Biodiversity at different levels. Number and range of different ecosystems and habitats, number of species, and their relative abundance, genetic variation within each species. Scientists often speak of three levels of diversity: species, genetic, and ecosystem diversity. In fact, these levels cannot be separated. Each is important, interacting with and influencing others. Changes at one level can cause changes at other levels. 18.2 3 Random sampling and biodiversity of an area. Random sampling in field work is important to collect data that is representative of the area being studied and to avoid bias in choice of samples. Random sampling is appropriate for habitats where species are distributed uniformly across the whole area and all are exposed to the same environmental conditions. 18.2 4 Different methods to assess the distribution and abundance of organisms in an area: quadrats, line transacts, Bell transacts, mark release recapture, Lincoln index. Frame quadrats are used to assess the abundance of organisms. Abundance can be recorded as species frequency, species density, and percentage cover. Line and belt transects are used to investigate the distribution of organisms in a habitat where conditions are not uniform, e.g. from low to high altitude. Line transects show qualitative changes in species distribution, belt transects show changes in abundance as well as distribution. Lecture notes for A Level Biology by Mohammed Rashid Chatha. 030046100457. 91. The Lincoln Index is a way to estimate the number of organisms in populations of motile animals in a particular place. Population size is determined by the mark, release, recapture method. 18.2 5 Spearman's rank, correlation, and Pearson's and linear correlations, biotic factors and abiotic factors. Spearman's rank correlation test is used to determine whether or not there is a correlation between two paired sets of data that can be ranked. When discussing an experiment, you need also to consider possible source of error and validity of the experiment, and be able to suggest ways in which the experiment's reliability could be improved. You should be able to reach a conclusion about the data and use of statistical tests, and perhaps make suggestions for further experimental work. The modified crop plants may become agricultural weeds or invade natural habitats. The introduced genes may be transferred by pollen to wild relatives whose hybrid offspring may become more invasive. The introduced genes may be transferred by pollen to unmodified plants growing on farms that grow organic crops. The modified plants may be direct hazard to humans, domestic animals, or other beneficial animals, by being toxic or by causing allergic reactions. The herbicide that can now be used on the crop will leave toxic residues in the crop which may harm consumers. GM seeds are expensive and their cost may remove any advantage of growing a resistant crop. Growers mostly need to buy seed each season, keeping costs high. In contrast, seed from this year's crop of traditional, non-GM varieties can be kept to sow for next year's crop. In parts of the world where a lot of GM crops are grown, there is a danger of losing traditional varieties. This is important because these varieties have desirable background genes for particular places and they may have characteristics that could be useful in a world where the climate is changing. This requires a program of growing and harvesting traditional varieties and setting up a seed bank to preserve them. Reproduction in animals. Common birth control methods. Name, procedure, methodology, effectiveness * action needed, risk. Vasectomy. Vas differentia are cut and no sperm in, almost 100% sexual, irreversible. Lecture notes for A Level Biology by Mohammed Rashid Chatha. 030046100457. 92. Tide. Semen freedom, sterility. Tubal ligation. Oviducts are cut and tied, no eggs in oviduct, almost 100% sexual freedom, irreversible sterility. Pill medication is taken daily shuts down anterior pituitary. Almost 100% sexual freedom thromboembolism. Norplant progesterone implant is placed under skin by physician. Shuts down anterior pituitary. 99.7% sexual freedom. IUD inserted into uterus by physician. Prevents implantation more than 90% sexual freedom infection. Sponge and diaphragm inserted into vagina at cervix. Blocks entrance of sperm into uterus. With jelly about 90% must be inserted each time before intercourse. Condom sheath is placed over erect penis. Trap sperm about 85% must be placed on penis at time of intercourse. Coitus interruptus, withdrawal. Male withdraws penis before ejaculation. Prevents sperms from entering vagina. About 80% intercourse must be interrupted before ejaculation. Jellies, cream, foams. Inserted into vagina. Spermicidal chemicals kill a large number of sperm. About 75% must be inserted before intercourse. Rhythm method. Determine day of ovulation by record. Avoid day of ovulation. About 70% limit sexual. Lecture notes for A-level biology by Mohammed Rashid Chatha. 0300 461 0457. 93. Keeping, testing by various methods. Activity 18.2 to 6 Simpson Index for Biodiversity. Simpson's Index of Diversity, D, is used to calculate the biodiversity of a habitat. The range of values is 0, low biodiversity, to 1, high biodiversity. Spearman's rank correlation and Pearson's linear correlation are used to see whether there is a relationship between two features. 18.3 Conservation. Conservation of an endangered animal species may involve captive breeding programs, in which viable populations are built up in zoos and wildlife parks. These programs try to ensure that the gene pool is maintained and inbreeding is avoided. At the same time, attempts are made to provide a suitable habitat in the wild, so that captive bred animals can eventually be reintroduced to the wild. It is important to involve local people in such projects to increase the level of acceptance and the chances of success. 18.3 to 1 Extinction of species. There are many threats to the biodiversity of ecosystems. These include climate change, competition, especially with alien species, hunting by humans, and degradation of ecosystems leading to habitat loss. These factors are responsible for local extinction of populations and the total extinction of species. There are moral and ethical reasons for maintaining biodiversity, and also more practical ones. For example, plants may be sources of future medicines, and animals may provide alleles to use in animal breeding. 18.3 to 2 Reasons for the need of biodiversity. Methods of assisted reproduction, such as IVF, embryo transfer and surrogacy, are used in the conservation of endangered animals. Frozen zoos maintain collections of sperm, eggs, embryos and tissue samples of endangered species in cold storage. 18.3 to 3 Roles of zoos, botanic gardens, conserved areas, frozen zoos and seed banks in the conservation of endangered species. Botanic gardens and seed banks help to conserve threatened plant species by breeding them for reintroduction into an appropriate habitat. Seed banks provide suitable conditions to keep different types of seeds alive for as long as possible. Lecture notes for A-level biology by Mohammed Rashid Chatha. 0300 461 0457. 94. Samples of the seeds are grown into adult plants every now and then, so that fresh seed can be collected. Many countries have protected areas called national parks, which often cover large areas. National parks are set up to conserve rare and endangered species and to maintain their habitats. Often legislation is passed to ensure their protection. Within national parks, activities such as agriculture, building, mining, and other industries are forbidden or strictly regulated. access may be limited but not forbidden, as one aim of most such parks is to educate people about the importance of conservation. Marine parks have been created in many parts of the world to protect the biodiversity of endangered ecosystems such as coral reefs. These parks usually prohibit fishing. One benefit of marine parks is that populations of fish in the area have increased. Other smaller conservation areas may be created to protect particular species and habitats. 18.3-4 Methods of assisted reproduction of species. ARTs include artificial insemination, in vitro fertilization, IVF, and cloning, amongst others. ARTs were originally developed for breeding animals like cattle, horses, or sheep for human consumption and use. These days, however, we see this technology being applied to a wider range of organisms. 18.3-5 Reasons for controlling invasive alien species. Alien species are those that have been introduced or have invaded ecosystems where they did not exist before. Alien or invasive species need to be controlled because they pose threats to native species. Alien species may be effective predators and/or competitors with native species. Non-governmental organizations, such as the Convention on International Trade in Endangered Species of Wild Fauna and Flora, CITES, and the International Union for Conservation of Nature, IUCN, play important roles in local and global conservation. Examples of barriers that lead to reproductive isolation of two populations of a single species. If sufficient genetic differences occur in the isolated population, new species are formed. Stage of life cycle at which barrier is effective. Barrier to interbreeding explanation. Before mating, geographical separation, populations inhabit different continents, different islands, or different sides of a large canyon. Habitat isolation, populations inhabit different local. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 0300 4610457. 95. Habitats within one environment. Temporal isolation, populations use the same environment but are reproductively active at different times. After mating, incompatibility of gametes, female might kill sperm of wrong genotype or pollen of wrong genotype fails to germinate. Hybrid, not viable, hybrid fails to develop to maturity. Hybrid sterility, hybrid grows to maturity but is sterile. 18.3-6 Role of IUCN for the conservation of nature. IUCN is at the forefront of the global fight to save species from extinction. IUCN experts, including over 8,000 species survival commission members in 130 specialist groups, and tools such as the IUCN Red List of Threatened Species trademark symbol, provide technical expertise and guide conservation action worldwide. Keywords: abiotic factors, abundance, alien species, animalia, archaea, bacteria, belt transect, biodiversity, biological species concept, biotic factors, classification, climate change, competition, conservation, culling, distribution, domain, ecological species concept, ecosystem, embryo transfer, eukarya, frozen zoo, fungi, habitat, in vitro fertilization, IVF, Lincoln index, line transect, mark release recapture, morphological species concept, niche, nucleic acid. Pearson's linear correlation, Plantae, Protactista, Quadrat, random sampling, RNA virus, seed bank, Simpson's index of diversity, Spearman's rank correlation, species, surrogacy, taxonomic hierarchy, virus, Worldwide Fund for Nature, WWF. Chapter Glossary. Abiotic factors are the non-living aspects of an ecosystem, e.g. light, temperature, water availability, oxygen availability, and soil pH. Abundance is the number of individuals per species in a specific area at any given time. Alien species is a species that is not native to an ecosystem. It may be introduced deliberately or accidentally into the environment. Animalia is one of the kingdoms of living organisms. Members are multicellular eukaryotes without cell walls. Archaea is one of the domains of living organisms. Members are single-cell prokaryotes with ether-linked cell walls. Bacteria is one of the domains of living organisms. Members are prokaryotes with ester-linked cell walls. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. 030046100457. 96. Belt transect is a line along a sampled area upon which quadrats are placed at intervals to determine the abundance and distribution of organisms in an ecosystem. Biodiversity is the variety of genes, species, and habitats within ecosystems or habitats. Biological species concept defines a species as a group of organisms whose members can interbreed to produce fertile offspring. Biotic factors is the living components of an ecosystem, e.g. food availability, pathogens, predators, and other species. Classification is the process of grouping organisms based on their phylogenetic characteristics or evolutionary heritage. Climate change is long-term changes in usual weather and temperature patterns. Competition is when different organisms compete for the same resources, e.g. light, water, mates, territory, in an ecosystem. This limits population sizes. Conservation is the maintenance of ecosystems and biodiversity by humans in order to preserve the Earth's resources. The conservation of species requires the preservation of gene pools. Convention on International Trade in Endangered Species of Wild Fauna and Flora cites a treaty that regulates the trade of plants and wild animals across international borders. Culling is separating of organisms based on whether they have desirable characteristics. Organisms with undesirable characteristics may be killed. May serve as a method of animal population control, including in conservation efforts. Distribution is the spread of living organisms in an ecosystem. DNA virus, a virus whose genetic material is DNA. This group is subdivided according to whether the DNA is single-stranded or double-stranded. Domain is the highest taxonomic rank. It is the broadest classification of living organisms. Ecological species concept defines a species as a group of organisms that are adapted to a particular niche. Ecosystem is the community of organisms, biotic and non-living, abiotic, components of an area and their interactions. Embryo transfer is implantation of embryos into a uterus during assisted reproduction, e.g. IVF. Eukarya is a domain of living organisms. Members are eukaryotes. Their cells have a nucleus which contains DNA. Frozen zoo is a collection of genetic material, such as DNA, gametes, and tissues, extracted from organisms and preserved at very low temperatures. Fungi, a kingdom of living organisms. Members are non-photosynthetic eukaryotes with chitin cell walls. Habitat is the region where an organism normally lives. International Union for Conservation of Nature, IUCN, an international organization that promotes the conservation of nature and sustainable use of natural resources through scientific research, management of field projects and implementation of legislation. In vitro fertilization, IVF, is an assisted reproduction technique which involves collecting male and female gametes, fusing them in the laboratory and stimulating embryo growth before embryo transfer. Lincoln Index, a statistical measure used to estimate population sizes of animal species. It is based on the mark release recapture method. Line transect a line along a sampled area. The species touching the transect at regular intervals are recorded to determine the abundance and distribution of organisms in an ecosystem. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. O304610457. 97. Marg release recapture is a method of estimating the population size of motile organisms. It involves capturing a sample of the population, marking them and releasing them. At a later date, another sample is captured and the number of marked individuals recorded. The population size can be estimated using the following equation. Morphological species concept defines a species as a group of organisms that share similar physiology and appearance. Niche describes how an organism fits into an ecosystem and its role in that environment. Nucleic acid is a polymer of nucleotides that stores genetic information, e.g. DNA or RNA. Pearson's linear correlation is a statistical test which determines the strength of the linear correlation between two variables. A value of 1 indicates perfect positive correlation and -1 indicates perfect negative correlation. Plantae is one of the kingdoms of living organisms. Members are photosynthetic eukaryotes. Protoctista is one of the kingdoms of living things. Members include any eukaryotic organism that is not a plant, animal, or fungus. Quadrat is a square grid of a known area used in sampling to determine the abundance of organisms in a habitat. There are two types, point quadrats and frame quadrats. Random sampling is a sampling technique used to avoid bias, e.g. creating a square grid and generating random coordinates. RNA virus is a virus whose genetic material is RNA. This group is subdivided according to whether the RNA is single-stranded or double-stranded. Seed bank is a storage of seeds to preserve genetic material. Simpson's Index of Diversity, a measure of biodiversity in a habitat which takes both species richness and species abundance into consideration. It can be calculated using the formula. Spearman's rank correlation is a statistical test which determines the strength of the non-linear correlation between two variables. Species is a group of organisms that can successfully reproduce with one another, producing fertile offspring. Surrogacy is one female individual carries the offspring of another female. Taxonomic hierarchy is the non-overlapping classification of living organisms according to their common ancestors. There are smaller groups within larger groups. In order from the largest group to the smallest group, domain, kingdom, phylum, class, order, family, genus, species. Virus is a non-living microorganism that consists of genetic material surrounded by a protein husk. Worldwide Fund for Nature, WWF, is an international non-governmental organization committed to the preservation of wildlife and reduction of human effects on the environment. Lecture notes for A-Level Biology by Mohammed Rashid Chatha. O304610457.